All right, let's get started. Morning, everyone. I'm Bin Chen. I'm a faculty at Michigan State University. My lab just moved to Grand Rapids from Silicon Valley early this year. So when I come here, I heard about this conference. I was so excited. I was so impressed by the big data community here in Grand Rapids. My lab, big data lab, just a few blocks away from here. So that's why there's no reason we don't come here. That's why I come here to present, and also my lab is here to volunteer. So thanks for the organizer to organize this fantastic conference. Today I'm going to talk about how we're going to use the big data and AI to discover new therapeutics. I want to start with one Chinese game. It's called Go. It's very complicated games. You need a lot of the tactics, and then you have to know your opponents. You have to make many steps ahead. Usually we think only smart people can play. I don't play, and I don't watch either. But I watched the game in 2016 because one player is a computer. It's called AlphaGo, developed by DeepMind, owned by Google. Another player is world-class Go player, Lee Sentong. Surprisingly, AlphaGo won these games. And later this year, it beat all of the world-class Go players. So this really get all the world excited, including myself. What's AlphaGo, right? Well, AlphaGo is just computer games. First, it does not know anything about the Go games, but it plays. Every time, either it fails or succeeds. It learns from the games a little bit, and then improve itself. Our computer does not like our human being. We need to take a break after the game, but computer does not take a break. It can train itself 24/7. So after numerous training, until nobody can compete with AlphaGo. Or AlphaGo itself is not really just a Go game, or just a player to play Go. It's really present the entire field, the AI. We think, well, whether AI started to boom. So what is AI? It's quite a buzzword. If you ask a different person, you would get different answers. My answer is that AI is just simply a set of intelligent computational programs that help our human being to address the challenge which we are not able or hard to address. It includes the conventional machine learning method and also the advanced like deep learnings. And deep learning probably is one of the reasons why AI becomes so popular. And it originated from the computer vision field. So in computer vision field, every year they organize competition. They invite the researchers across the world come together to make competition and just to develop the method to recognize the image, whether it's a dog, cats, cars, right? So before 2012, the performance around 75%. It's just so, right? But in 2012, there's one team stand out. It performs much better than others. People start to try to understand what's this, right? And they found out this one group from University of Toronto is using one method called deep learning. Later, after a year, every groups trying to use deep learning, and today it performs as good as a human being. And the competition discontinued because there's no reason to compete, right? So deep learning is really just a deep neural network. It composes many layers. In the input layer is image, like cars, cats, dog. The output layer as different labels, either it's car and this truck, van, etc. The middle layer is many composed by many we call the neurons. It's like neuron in our brains, right? Our brain essentially this method trying to mimic how the brain function. And when we are a child, we learn the world by watching, right? We watch lots of objects, right? It takes many years for our brain to develop. Same as deep neural network, it also needs lots of data to train to understand the image. And deep learning, this method itself is not new. The reason it started booming because we have too much data. We have millions of labeled image to train the model. We have tons of the computational power. So we have thousands of CPU can work together to make predictions. Today, the technology is simply explode. It can use a computer to generate the figures. These are cats. They are not actually the picture of the real cats. It's just you send us a random number, the computer synthesizes the cats for you. If you upload the image, the pictures, the computer can convert the image to the style of different artists, like the Monet, Van Gogh, you name it. Well, computer even can help you dance. Even you are not a professional dancer, you don't dance ballet, but don't worry, the computer can help you. At least it can make a video looks like you can dance ballet. Right? Well, this is just the key technology behind. Of course, the impact is profound. You can think about this video, right? I bet in the future, you would not trust the evidence from the video anymore, right? And indeed, the computer, the AI is changing, reshaping every field, from banking, business, healthcare, transportation, etc. Give you one example in transportation. Because of the image recognition method, now we get a tremendous improvement. We can precisely recognize which is person, car, motorcycle, helmet. This really help, the computer. We can help to self-driven, right? That's the core technology we can, the self-driven car have to rely on. The computer can also help the doctor to diagnosis. This is just one study published last year. The computer can recognize malignant skin cancer from malignant skin cancer, or not malignant cancer from non-malignant skin cancer. And it performs as better as dermatologist. So which means the computer eventually can assist the doctor to make decision. Just within a couple of years, you will see lots of application using AI is computer to help to recognize the imaging in different disease field. The AI-related technology already went to the clinic, benefit the patient. Just a couple of years, the FDA already approved a few AI-based softwares. This is just one software called IDXDR, which used to detect diabetes retinopathy. On the right is another AI-based products to detect wrist fractures. If you are in Silicon Valley, you will see tons of the startup funded using big data and AI to address the challenge in the biomedical fields. So my lab, we are interested in how we can use big data and AI to discover new therapeutics, especially cancers. In the past decades, we made a tremendous progress to identify new therapeutics for cancers, but still, lots of cancer, we really don't have any treatment at all. That's why the cost of treatment is very high today. When you look at this bar, by average, the patient have to pay over $10,000 for the cancer treatment. $10,000. So you can imagine how many families can earn such amount of money. So my lab, we think, well, maybe we can use big data to help. Maybe we can use big data to reuse existing drug to fight deadly cancers. So speaking of big data, how big is it in the biomedical domain? I just give you a quick example. In 2014, European Bioinformatics Institute hosts 25 purpose data, and it quickly increased to 60 purpose data in 2015. So what does that 60 purpose data mean, right? Everyone has a laptop. That's about 2 terabytes storage. So 6 petabytes equal to the sum of the storage of over 30,000 laptops. 30,000 laptops. So you can imagine how much information can be stored in this laptop. Indeed, European Bioinformatics Institute hosts 120,000 data set. 120,000 data set. So what's the big data look like? I just take one example in liver cancer. We have the image data, like MRI, pathological image. We have the clinical data, like medication, outcome, the lab test result, etc. We also have the molecular data from the patient, like their mutation, copy number variation, protein expression, etc. We also have the emergent data set, like the single cell data. You can look at the individual single cells to look at the molecular features. We also have the microbiome, like from your gut, etc. In my lab, we are particularly interested in one type of data. It's called mRNA. Based on the central diagram, I think mRNA has much more information compared to DNA because it's more functional related. And compared to the protein, it's more stable. And more importantly, we have tons of data for the MR, which can be used like using, originally can using microarray, now using RNA-seq to get. Just one example in GEO, it's the one public gene expression repository. Now it includes over 2 million samples. 2 million samples, majority of them are bulk tissue. Now if you look at a single cell, look at individual single cell, now we have over 500,000 single cell. In 2014, you can publish a paper in Science using just 400 single cells, but today you need 100,000 single cell to be able to publish in Science, right? So what information in this like 1,000 single cell? We just can give a quick calculation. Suppose we have over 500,000 single cell. Each single cell has 20,000 features. So in total, that's about 10 billion time points, 10 billion data points. Now, you know how many single cells in our body? That's trillions of single cells, right? If you times 20,000 features, that's a lot. And this only capture gene expression. If you want to look at the protein expression, metabolic expression, that's just too big. Yeah. So how are we going to use this data, right? What's information inside this data? It's just simply a letter, ATCG, ATCG. But we use this data, we can detect whether a patient is affected a lot, whether it's affected by virus or bacteria. We can also use the data to identify small molecule or drug to treat the patient. We can even use the data to predict whether it's a patient or to respond to like immunotherapy or not. Let's just show you one example. It's called ICE-BY trial. It's called International Clinical Trials for Breast Cancer Patients. I was fortunate to be part of these programs. It's a very complicated diagram, but just want to show you every step we treat the patient based on their molecular data, like biomarkers. If the patient has HER2 overexpressed, we're trying to find HER2 drugs for this patient, right? If it's ER positive patient, we're trying to treat with the ER related drugs like taximoxifen. But if the patient doesn't respond, we try to come out with some AI algorithm to recommend the treatment for that patient. So you can see it's very complicated treatment protocols. But just want to tell you, today we already be able to use this molecular data to guide therapeutics in the clinic. So just show you a few examples how we're going to use this to in the research. So for example, if we want to understand the disease mechanisms, right? We want to know with this liver cancer, what's going on wrong in these tumors, right? We can take out the tumors and profiles. But it's not enough, right? You want to know which part goes wrong, which genes go wrong. You really need a controls. So most of the times, we just took a little bit tissue samples adjacent from the tumors, right? Then we can use that control to be able to create disease signatures. We present, which genes are abnormally upregulated, abnormally downregulated. But if you look at the database like TCGA, you will find half of the cancers we don't have enough adjacent normal tissue samples. Why? For example, in brain cancer, right? The patient or a lot want to donate their adjacent normal brain for the research, right? The good news is that there one project called GTEx provides the profiles of thousands of health individuals. It's supposed to be a good resource as a reference. Now, the problem is coming. This database, they're using different platform to process or profile their samples. And they're quite different, right? So how can we use this as a normal? Can we compare the normal to the tumors? And you can consider each sample as a set of features, many like high dimension features. And if you compute the correlation between tumor and normal, you think, oh, you just simply let's just look at a Pearson correlation, right? Or you want to do some PCA, do the dimension deduction. But PCA usually does not capture the nonlinear relationship between each features. So we decided to explore the deep learning approach. We're using deep learning autoencoders. Here are just one very simple architectures. Instead of using image data, we're using genomic data, like 60,000 features. The output also is the 6,000 feature. And we're trying to use the autoencoder to learn this sample, like 20,000 samples, and find the best model to be able to represent these features, right? And this is a very standard approach, and we compare with the PCA and others common approach, and found out the deep learning approach performs much better. This is just one example in breast cancer nomina A studies. We compare the breast cancer lumina A cancer samples with all the normal samples from GTEC. Like all of them are from the health individuals. And you can see half of the normal samples from breast correlated to breast cancer lumina A, but a significant number of the normal samples are not correlated to breast cancer lumina A, even they are from breast. So which means even we are using breast as a control, we cannot use every tissue samples as control. You have to classify the tissue samples as well. This just tells you that we need deep learning or other method to help. Then how are we going to translate these data points into therapeutics, right? We think if a disease is a molecular disease, there is a molecular signature for that disease, and we can find a drug to correct that signature. The simple idea is that you can compare the disease, the profiles, like RNA profiles of the disease samples and control samples, create a disease signature. Here, the red can show some genes abnormally upregulated in the disease sample. Some genes are abnormally downregulated in the disease samples. We can also treat the cells with the drug and without the drug to get the drug signatures, right? Because we are using cancer cell line, we can get a signature for all the FDA-approved drug or drug in the clinical trial. Then we can match the disease signature and drug signature and see any reversal correlation. Which, if we think the drug can reverse that disease signature, then the drug may have some therapeutic effect. The idea is very simple. We'll just show you one example in HCC, hepatocellular carcinoma, which is a very common liver cancer. The idea is quite simple. We just took out the tumors and the adjacent tissue samples and create a signature for consisting of 274 disease genes. So some of the genes are abnormally upregulated, some genes abnormally downregulated. Then we use this disease signature to query our drug libraries. Here is the first column, shows the disease signatures, HCC. Remaining column are the drug signatures. And red shows the upregulated genes. Green shows the downregulated genes. And if we want the drug to reverse that disease signature, we really want the red color goes up, green color goes down. And you can see the majority of the compounds can suppress, can reverse disease signature, except the last one, which we just add on purpose. But surprisingly, there's one drug called niclosamide can reverse the disease signature. Niclosamide, you know guys, what's this drug for? It's a drug to kill the parasite in our body. But in the USA, people rarely get affected by this disease, so you rarely see this. But in a tropical place, the kids have to mandatorily take this drug to kill the parasite. Then we think maybe this drug can be used to treat cancer, to treat the liver cancer. Then we go to the lab and test that. And we find that, surprisingly, this drug, the salt of this drug can significantly inhibit tumor growth. This just shows you this liver, and this is the tumor, and you barely see the tumor and the treatment group. And this is another models. This is the control, and this is the treatment groups. And we compare our drug with the standard of care sorafenib. And this is the group, the mice just treated like water and regular food. And this is our drug, and this is the sorafenib. And you can see our drug is effective as sorafenib. And the combination has much more profound effect. You barely see the tumor in the two mice, which means if the liver cancer can take our drug and sorafenib to be able to get I don't think we can get a cured, but definitely can slow down the disease progression. Again, we just show you we're using big data to find a potential therapeutic candidates. We just show you one more example in Ewing Sarcoma, which is pediatric cancers. The idea is quite similar. First, we also create a signature for Ewing Sarcoma by comparing their tumor samples and the normal samples. And second, we know this Ewing Sarcoma is driven by the Ewing genes. The Ewing, if we lock down of this Ewing gene, can reverse the disease phenotype. Then we think, well, maybe you can find a drug to mimic the siRNA of Ewing genes. We also look at the compounds which can overcome the drug resistance. And then we make a model and we predict 28 compounds using the different models. And we send it to our collaborators. They test these compounds and found out 17 of them are positive, which means their success rate is over 50%. So if they go to the lab, do the high super screening, that's the conventional approach. That's 5%. Our success rate is 50%. And of course, approach is very cheap, right? You are using open data, you just need a laptop. And very fast, right? We just build a competition model. But if they run the screening, they take a few months to set up all the experiments. So this really shows our approach has unique advantage for the drug discoveries. This is another example. In basal cell carcinoma, it's one type of skin cancer. In this case, we first build a signature for advanced basal cell carcinoma patient, and also build a signature for early stage basal cell carcinoma patient. Then we predict drug for each type of the patient, and then come out one common drug, which is the varenstat. It's HDAC inhibitors. And we found that the HDAC inhibitors can suppress the BCC growth in vitro and in vivo. Vivo means the animal models. And also they can reverse the basal cell carcinoma gene expression signatures. We get lots of success in several other diseases. This really motivates us to think about why this approach works again and again. Then we did a big data analysis. We analyzed tons of data, tons of cancers. First, we quantify the reversal correlation. We called potency to reverse disease gene expression. Then we pull out the drug effects data from different labs. So each lab, some lab tests this group of drugs, some lab tests that group of drugs. We pull them together, and then we found the interesting correlation that the potency to reverse disease gene expression correlated significantly high with the drug efficacy, which means if the compounds show high potency to reverse disease gene expression, more likely these compounds are effective in a cell line, in a virtual validations, which means we don't have to do experiment, right? We get a prediction. Most of the times, the drug will be positive. We also see the pattern in breast cancer, colon cancer. Well, if you guys are data scientists, you may say, oh, this is not perfect, right? It's not like a linear correlation. That's fine, right? We don't care about like AUC, ROC. We really just need one drug, right, for one cancer. So the best way is just go to the lab, use our models, make predictions, and test in the lab. So in this liver cancer, we use our approach. We scored over 12,000 compounds, and we ask our collaborators to select a few compounds which are novel in liver cancer, and also represent different mechanisms of actions. And they select the 4 compounds, and they test in 5 liver cancer cell lines. On the right just shows the efficacy of these compounds. Usually, the smaller, the better, right? And if it's around 1, which is good. But if it's less than 1, which is perfect. Your collaborators all love it. And we are excited that our compounds, one of our compounds, their IC50 value is very small, and then they test this compound in the animal models, and they found that the compound can significantly inhibit the tumor growth. So this just shows you this approach can use to predict the drug efficacy in vitro and in vivos. So my lab now we are actively improving and implement this approach in different disease. We can look at a primary cancer, metastatic cancer, or trying to understand the drug resistance. Or we can look at a single cell, see how this drug can regulate the single cell activities. In terms of the molecular data, we not only use gene expression, we can also use protein expression, metabolic expression, right? Regarding the signatures, drug signatures, now we are using cell line to get the drug signature. We can also use the signature from more advanced models like organoid, animals, etc. I'll just show you a couple of examples. Last year, my lab, we had a very crazy idea. We think, can we predict drug for every cancer, like every cancer? So we decided to like crazily integrate all the cancer samples, and we use the same pipeline process their samples. And now we have over 20,000 samples cover like over 40 cancers. Because our deep learning approach I presented earlier, we can use that to select reference normal tissue samples. So theoretically, we can predict drug for each cancer. Well, if you have your own samples, we also have this pipeline to be able to process your samples and integrate into our systems, and allows us to, to predict the drug for the samples you have. We also look at the single cell data. My postdoc, Dr. Ramashankar, is behind. He's interested to using single cell data to be able to predict drug for the individual patient. So given one patient, we have their single cell data. Then we can create a signature for that patient, and we can predict one drug or multiple drugs to treat this patient. We also developed the portals. We have two versions. One is a desktop version. If you are a computational biologist, you can use our very advanced versions and can change the parameters, predict drug for the cancer you are interested. If you are a bench scientist, you can go to our web portal and just select the cancers you are interested in and specify the features, and our system can automatically retrieve the samples from public database and create a signature for that particular disease. They make a drug for that. Well, we're still actively developing this portal. If you guys are excellent developer, feel free to contact me later on. So maybe majority of them are data scientists or software engineer. You have no idea. I am not a scientist. I don't know drug at all, right? We need to test, run many experiments to test our hypothesis. But don't worry about that. Today, someone can help you. So for example, the company, they can provide service. They have 60,000 service. From the very beginning, there are in vitro screening to a very advanced animal validation. They can help you as long as you have enough cash. But I don't think it's very expensive. My lab, we are not only interested to repurpose an existing drug, but we also think about how we're going to develop much better drug, right? So in our approach, we really need a very good profiles. Like there are gene expression profiles. We need to treat the cells with the drug to get the profiles. But that's very expensive to get the data. Think about it, we have millions of compounds. It's just impossible to get the profiles of all these compounds, right? Then we decided to think, well, maybe have to using some AI approach to help us to predict the profiles of a given compounds. But we know these profiles, they really depends on the chemical structure, the cellular context, the biological conditions. The good news is that we have tons of data available. We have close to 500,000 drug profiles under different biological conditions. And we know if you are using conventional linear regression, it's just impossible to make predictions. So we can think about whether use deep learning to help. So that's one of my postdocs is working on. And this just shows some results from other students. So we're trying to use deep learning again to synthesize the chemical structures. And the right one, that's the chemical structures. And the left is at the very beginning, it says there it's blur, right? But as the model started to train, you see this looks like chemical structure, right? But of course, not perfect. We are still working on this, optimize this this method. Hopefully, we can just use the deep learning to be able to find brand new chemical compounds. We're also interested in many other fancy technology. This just show you one examples. On the left is do you guys know this? This is called deep lens. Amazon recently just released this. It's a very intelligent camera. Basically, you can deploy the AI into this camera. And this can help you. You can just use the camera and just look around to the basic stuff. This just show one demo from my lab. You just simply use this camera can detect. This is a TV monitor. This is dog. What's the probability to be a dog, etc. Of course, we are not interested in dog, cats. We are interested in patient, right? So we want to say, well, maybe we can replace this dog with the patient. See how we can use the deep learning to help the patient. Yeah, I'm very excited. Now we have fantastic teams with the same passion and interest. And we're also looking for talents. If you're interested, just let me know. We can talk. And I would like, again, thank the organizer for this conference and all of my collaborators help us, and also my funding resources. I'm happy to take any questions. Yes, that's very important because we saw that if you think about the chemical compounds, every compound is toxic, right? Increase the dose, you can change everything. So of course, dose is very important to determine the profiles of these compounds. We have to consider that into our modeling. Yeah.
Okay, so you did take that into account. That was one of the variables that went into the AI.
Yes, we did. Yes. Yeah, that's a good question. So we are using open data. So basically they're using very standard dose, like 10 micromolar in their treatment, or 6 molar sometimes, depends on. But majority of the 10 micromolar. Ideally, we want to change different dose and look at how the drug behavior. But because no model, no data is perfect, right? At the end of the day, we just use our model help us. Hopefully it can save us time and efforts to run experiment. We can use the model to prioritize compounds. At the end of the day, we have to go to the lab and test under different dose and different models. There are many issues like PK, PD, toxicity issue we have to take care of. Hopefully we can build a streamlined process. Every step we can build a model to guide. That's one of our goals in my lab. Yeah, so that's one of why we developed this portal. The conventionally have to test compound each compound in the lab. Now we have the profiles of the compounds. We know which genes are going to be changed after treatment of the drug. Then we can match their profile to the drug signature and see whether this compound can reverse. Based on their reversal scores, we can prioritize this compound and choose this compound which will show like high probability to reverse that disease signatures, from which you can select the compounds. Yeah.
models actually used for ACC?
Yes, yes. So we look at thousands of FDA-approved drugs, pretty much all the FDA-approved drugs. We just score them and rank them, and then we found niclosamide is one of the best to be able to reverse HCC signatures.
Based on the molecular consortium?
Yes, yes, yeah. And we also get a success in many other cancers. As I showed, Ewing Sarcoma, basal cell carcinoma, and recently we even got a success in Alzheimer's disease. It's non-oncology, yeah.
So I'm curious, is it possible to then reward your model, reward your AI with, even like a bigger win? It found this compound and it's actually effective now. Feed that back in, augment your model so that the next time you go in,
Yeah.
you can learn from its success there.
Yes, that's exactly this project going. We wanna. So we actually started. So we first design a group of compounds, then test. We get the efficacy. Then we go back, optimize our model, design another bunch of compounds, and come back test. It's more like the AI. Very beginning I show you AlphaGo, but it's just take a while. They take maybe a couple minutes. We take a couple months to get the feedback whether these compounds are effective or not. But the essential idea is that we wanna use our model we call it semi-supervised learning. To help us. Yeah, everything. This, I think, is a semi-supervised learnings. Yeah, but it was they call it reinforcement learning. But I think that's very advanced. Yeah.
Just curious, just because we still have some time. So what is your take on, the science being where, since we're collecting all this data and we're testing on animals, there was another talk where they were from View, where they were actually testing on fish. Here I saw a lot of slides with rats, but we're collecting all this data, right? Do you see science going to a place where we actually don't have to test on animals or creatures, but rather we have all this data and then we can actually play around and then predict? Or I'm just asking for your general take on this.
Yeah, I think that's the goal. We wanna build tons of the model. Eventually, we wanna use this model. Hopefully, it can replace some animal models, or even can synthesize like the patient. When you run the clinical trial, now it's very expensive to recruit the patient, right? And we need a good model to help us. Yeah. But now we still need lots of data from the cell line, from animal model to be able to create a robust model to make predictions. Yeah. Good. So we have pretty early. Well, we're 5 minutes early, but it's good. Thank you.